Chapter 16 is an introduction to cancer genetics. Cancer cells are characterized by several things. Number one, uncontrolled growth. They cannot stop themselves while going through the cell cycle. Now, most normal cells can stop at checkpoints if something's going wrong, but cancer cells can't, so they keep growing and growing when they shouldn't. Number two, they're characterized by the loss of what we call contact inhibition, or you might have learned it as density-dependent inhibition. And what that means is when normal cells grow on a plate, when they get a certain density or crowdedness, they'll stop growing. It will send a signal into the cell to stop growing, and they'll go into a G0, a resting state. But cancer cells don't have that ability. They keep growing even though they get too crowded and they start to pile up. And that's the equivalent of a tumor. Number three, they accumulate a lot of gene mutations and large chromosomal abnormalities like deletions, duplications, inversions, and translocations. And that's partially the result of the fact that they're unable to stop during the cell cycle to repair anything that's going on. And number four, and maybe the most important characteristic, is the ability to metastasize or spread to other parts of the body. And so here you have a picture of a human liver that has lung tumors that come from lung cells. And you can see here, these are obviously not, in their original location. So when you have a lung cell in a liver, it means that those cells have traveled. And you start off with a tumor in the lung. Some of those cells are able to get into the bloodstream or into the lymph system, and they can then seed new tumors in other places. This is a karyotype of a cell that is cancerous. You can see a lot of the chromosomes don't have homologs. Well, they have homologs, but they're up here in this pile. These are the derivative chromosomes that are basically the result of translocations, fusions, deletions of those other chromosomes. So up here at the top, I don't know if you can see it, but it's pretty faint, but these numbers represent the chromosomes from which these new chromosomes are derived. So this is a piece of chromosome 9 fused to a piece of chromosome 1. You can see that here, and so on. So you have this mess of chromosomes here that are all large chromosomal abnormalities. And so this is pretty typical of a cancer cell. Its karyotype is just a mess. From birth to death, your chance of getting some type of cancer is 1 in 2 or maybe 1 in 3. It's a major source of mortality among humans. And here's a list of, different types of cancer are more common than others. Prostate cancer being fairly common, but not as many deaths per year. Some things are more treatable or more successfully treated than others. Now, most cancers are sporadic, and people probably don't understand this, but most people who get cancer, they're the only one in their family or they got that type of cancer. And so that's just a sporadic, just means they got a mutation in one cell, in one gene, and that triggered that cell to become cancerous, or it started maybe a process that eventually led to cancer. Those with an inherited component only make up about maybe 10%, 5 to 15% of the cancer cases. Now, if you have an inherited component, you have to be very careful and get the correct screenings done, because that 10% of individuals, have among those individuals who have an inherited component, they almost always get that cancer. But it doesn't make up all the people who get that kind of cancer, because a lot of the cases are sporadic. And most cancers result from an accumulation of several gene mutations in the same cell, and we call that the multi-step model of cancer. We'll look at the first model of cancer we're going to look at is the simplest one. It's a two-step model. And we'll work our way up to the multi-step model of cancer, which we will look at with colon cancer. So retinoblastoma has just 2 mutations that lead to cancer, and that's a cancer of the eye. But then colon cancer has 4 to 5 mutations that lead to cancer. So those will be our examples that we use. Tumors are clonal in the same way that a bacterial colony is clonal, meaning you start with one original cell that gets all the mutations that are necessary in order to start the uncontrolled proliferation, and then as those cells go through the cell cycle and divide, you end up with a whole bunch of cells. That's the tumor that originated from just one cell. So we're going to talk about retinoblastoma first. It's a childhood cancer of the eye, and it's the result of 2 mutations, but they're 2 mutations in the same gene. So if you get a homozygous mutation in one cell, in one gene, the gene is called the retinoblastoma gene, then that cell will start to grow uncontrollably and create a tumor in the eye. And here you can see some children that have this. I just took these off of the internet. But you can see with the 2 children on the left side, there's kind of an unusual glare, but that represents where the tumor is, although you might just mistake it for a weird flash on your camera. But on the right side, I don't think that there's any question about what is going on with these children on the right. So in 1971, Alfred Knudsen, who was working at M.D.Anderson in Houston, and you'll know that M.D.Anderson is still a premier cancer institution in terms of research, he proposed what we call the 2-hit hypothesis as the cause of retinoblastoma. So there have to be 2 hits or 2 mutations, and it's 2 mutations in the same gene, the retinoblastoma gene, which is just called RB. And so if a single retinoblastoma mutation is inherited, then all the cells in that individual's body have 1 mutation. That's the first hit. Then if just one additional cell or one cell in the retina gets a second mutation in the same gene, then that cell will become the first cell that will form then the tumor. But if no retinoblastoma mutation is inherited, then you would have to have one cell that would accumulate a mutation in both copies of the same retinoblastoma gene in order to start the tumor. All right, so the rule is both the retinoblastoma genes must be mutated in the same cell in order to start the tumor. And so in this case, at the bottom here, you see if a person inherits one mutation, and they're showing the mutation as a little black dot here. If a person inherits one mutation, then all their cells have one mutation. And then all they need is a second mutation in a somatic cell on the other good copy of the retinoblastoma gene, and that cell will then start the tumor. If you don't inherit any mutation, it is possible to get retinoblastoma, but you would have to get the first mutation, and then in that cell or in one of the descendant cells, you'd have to get the second mutation. So you end up with a cell that has 2 mutant copies. That will start the tumor. So familial form of retinoblastoma, which is the one shown at the bottom, will typically result in early onset, childhood onset, and it will be bilateral, which in this case means affects both eyes. So you'll typically have a child that has tumors in both eyes. Okay, so that would be the inherited form, where you inherit the first mutation and the second mutation is sporadic, and that starts the tumor. Now, a totally sporadic retinoblastoma, in which you have to have 2 mutations sporadically in the same cell, that would tend to have a later onset. So, an older individual, not a child. And it typically results in a unilateral tumor, meaning only one eye. However, most cancers aren't caused by mutations in just one gene. Most types of cancer follow a multi-step progression with mutations in many genes. So this figure is showing the concept of the multi-gene progression, the multi-step progression. For example, you start with a normal cell, the cell's dividing, it keeps dividing, and let's say one cell gets a mutation, a sporadic mutation in a gene, and that cell starts to grow more quickly. But it's not cancerous yet. I mean, it is and it isn't. It's precancerous. Then that cell, as it keeps dividing, one of its descendant cells gets a second mutation. And it starts dividing even more rapidly, and maybe it's not repairing its DNA anymore. And then a third mutation could be the result of it not repairing its DNA. Maybe a third mutation causes it to undergo some structural changes, and it's still dividing very quickly. And a fourth mutation causes it to become malignant and spread out of the original location to a new place. So that's the multi-step progression. But what's interesting is all the pink cells here are kind of the sort of the tumor. So the first mutation starts the cell dividing rapidly, and those cells keep dividing rapidly and start piling up. But within that clump of cells, one cell gets the second mutation. And so this subgroup of cells within the original tumor starts to proliferate even faster. And from within that group, some of the cells within that tumor start looking even weirder, and some of those get, the final mutation. So within the tumor, you'll have sort of a spread. Some of the cells only have, one mutation, and they're just a little bit weird. But if you look through the tumor, you'll see, areas that represent growth from a cell that accumulated an additional mutation, and so on. And so you only need one cell in that whole tumor that accumulates, all the mutations, and that cell can then spread to other parts of the body and just become the source of the entire problem. So what kinds of genes, when mutated, cause cancer? Well, we have several types. We have genes that are important for DNA repair. Remember, xeroderma pigmentosum is a defect in DNA repair that's related to or results in skin cancer at high levels. Genes that are important for regulating the enzyme telomerase, or the gene for telomerase also. Genes that promote angiogenesis. Angiogenesis is the growth of new blood vessels, which typically a tumor, if it gets large enough, requires new blood vessels to grow into it in order to feed it. And genes that are needed for proper cytoskeleton or extracellular matrix structure. But these are all typically some of the genes that are part of the multi-step progression. The primary genes that sort of start the process are typically two types, either tumor suppressor genes or what we call proto-oncogenes. So we will go to, these will be the two that we focus on in the next lecture segment. We'll look at what are tumor suppressor genes and what are proto-oncogenes, and that will be our focus. But there are some other genes that can be involved. And I did want to make the connection back to the xeroderma pigmentosum because you're familiar with that. But we will focus on tumor suppressors and proto-oncogenes in this chapter.